So now let's move on to the third talk, which is also the last talk for this session and probably the whole conference. Let's welcome Patrick Zhang, who is a researcher volunteer from Azadeh Yazdan's lab from University of Washington. He's going to talk about does functional connectivity reflect an anatomical connectivity, a simulation study. Okay, so Patrick, just take it over.
Okay, cool. Let me present my screen here. Okay, awesome. Yeah, thank you for the introduction. My name is Patrick. I'm a research volunteer at the Yazdan lab at the University of Washington. And this is actually my first talk at any science conference, so thank you for giving me the opportunity. And today I'll be talking about whether functional connectivity reflects anatomical connectivity and how we used a network simulation to study this relationship. Sorry. So first, what is anatomical connectivity and why do we care? Well, if we're interested in the connectivity between two regions of the brain, A and B, one way to define this connectivity is by looking at the synapses from neurons in region A to neurons in region B. This is called anatomical connectivity, and it's probably pretty challenging to calculate in an experimental setting. So researchers often are interested in functional connectivity as well, which tries to measure the strength and relationships of dynamics between the two regions. Functional connectivity can be derived from some kind of brain signal, such as local field potential or LFP. And there's multiple types of metrics we can use for functional connectivity. And so in our study, we're interested in how each of these metrics reflect the underlying anatomical connectivity of the brain. So we're using a network simulation to study this, specifically a network model of a cortical chunk. And because it's the simulation, we have full visibility into both the positioning of neurons and synaptic weights between neurons. And so we can use this information to derive anatomical connectivity. And we also wanted to record local field potential from our simulation and use this LFP to calculate different functional connectivity metrics and then relate that back to anatomical connectivity. So our model is 1.5 by 1.5 by 2.6 millimeters in geometry. We're simulating on the order of 100,000 neurons, and we're recording LFP from 54 recording sites laid out as 3 by 3 grids on the right. And we're simulating for 40 seconds and using a tool called Vertex for both the modeling framework and the initial model parameters. And we're running our simulation on the Neuroscience Gateway portal. So once we had our model, we wanted to validate that the behavior of the model was relatively biophysically realistic. So I'll briefly go over some of the checks that we did. So first is we looked at a spike raster plot of all of the neurons in the model, grouped by their cell type and their positioning within cortical layers. So the horizontal change in colors in this figure represents a different neuron group, totaling 15 different neuron groups. Here we just wanted to check that the firing dynamics per group were different. And then for a single neuron group, we wanted to check that the firing rate histogram was a relatively log normal distribution. And then for a single neuron, we wanted to check its interspike interval histogram. And here we were looking for a Poisson distribution. Because our model is generating LFP, we also wanted to verify that the LFP generated was realistic as well. So here we're looking at a power spectral density curve of an LFP recording from the center electrode on the surface of the model. And we're looking for a 1 over frequency curve here. You can also notice that there's a bump around the gamma frequency at around 40 hertz. This is an artifact from our initial model configurations, and we're actively looking at how tweaking these configurations can change the size of this bump. So next, we computed a few connectivity metrics from our model. So for functional connectivity, we looked at coherence, which is a pretty common metric in neuroscience. Pairwise Granger causality, because that introduces a metric with directionality. So A's connection to B might not equal B's connection to A. And then partial Granger causality, because that introduces conditionality as well. So if we're interested in A's connection to B, it tries to account as much as possible for any other node C that could potentially be contributing to A and B's connectivity. And then for anatomical connectivity, The way we calculated this was by looking at all the synaptic weights in the model, inversely weighted by the distance from respective neurons to the recording sites. So I wanted to share some really preliminary results with the disclaimer that there's a lot more analysis to be done on both the model side and the connectivity metrics side. But one of the things we were interested in is the correlation between each of these functional connectivity metrics to underlying anatomical connectivity. And we're looking at this per frequency. So per frequency value, we have a correlation coefficient from each of the connectivity metrics to anatomical connectivity. And this correlation is calculated across recording sites, pairs of recording sites in the model. So a few interesting things we noticed. Partial Granger causality, the red line in the graph, seems to be performing the most consistently across frequencies. Both of the Granger causalities seem to be outperforming coherence at higher frequencies, specifically higher than 150 hertz. And there seemed to be a dip in all three of the correlations at around 40 hertz. If you recall, this is also the frequency range where we saw a bump in LFP. And so our current thinking is that whatever mechanisms in the model are causing that bump in LFP could potentially also be contributing to this dip in correlation. So for next steps, we want to investigate whether tweaking the model parameters affects the relationship between functional and anatomical connectivity. Specifically, if we tweak the parameters to get rid of that bump in LFP that we saw, does that also change the correlation at around 40 hertz for our metrics? There are additional functional connectivity metrics that we're interested in and other ways of calculating anatomical connectivity, such as only looking at excitatory or only looking at inhibitory neurons. In the longer term, we want to extend the model to simulate stimulation at certain sites, and then also study effects of potentially adding a plasticity rule such as STDP in the model. So I wanted to thank everybody at the Yazdan lab for giving me an opportunity to work on such a cool project. Professor Yazdan and Professor Shea Brown for their guidance, and my co-author Julian for all of his mentorship. Thank you very much.
Okay, let's thank Patrick for giving such a great talk. I think it's very interesting. But it's probably a short talk, so there are a lot of technical details that are hidden from the slides that I'd like to know more about. I guess people from the audience might have similar interest. Since right now we don't have a Q&A, let's just kick out with a question that I have. I'm just curious, can you briefly talk about what kind of model you build to do the simulation? Like, do you have that kind of info? Like, what kind of model you build? How you like basically. My understanding is when you have a neural network in theoretical neuroscience, you kind of have a bunch of connections and which are represented in W. But these W are kind of given weights, especially when you try to do simulation. And given the structure you build, how you got the functional information that is different from the structure connection that you build, because usually when the weight is strong, then presumably your functional signal should be strong. That is very correlated with your anatomic connection. So how is that true, or it's actually different?
Yeah. So a few things about the model, like the one unique thing. Well, so we're using a modeling framework called Vertex to do all the modeling. The one unique thing about that is it's able to generate LFP from the model. Specific to like the questions about like the characteristics of the model. So basically you set up the types of neurons that you want in this model, and like where they are lying, like where their positioning is in terms of the cortical layer. And then connections across neurons are calculated. Let's see, I have a slide here. They're calculating by using a 2D Gaussian spatial profile to model decay of connection probability. And they're also calculated like per groups of neurons in certain groups to connect it to other neurons or certain groups are also have different like parameters for this Gaussian spatial profile. So yeah, that's, I don't know, does that answer your question?
Yeah, I think, yeah, that's kind of more complicated than I expect with this tool, I guess. And just another minor thing, and I'm curious, do they also have the kind of the mechanism with like the ramping up, like the potential spike and all this kind of neural mechanism existing in single neurons that
Yeah.
really mimic the real physics process and triggering this LFT and spike trains?
Yeah, so the
Okay.
spiking model they use is an adaptive exponential integrate and fire model. So my understanding there is like it's an integrate and fire model, but it like because it's adaptive exponential, it's able to generate a lot more different firing patterns. And then like the activity is driven by like random currents into each of the neurons, and that's modeled as a Ornstein-Uhlenbeck process. And so we, as the users, we configure the mean and standard deviation of that process.
I see. Cool. Thanks for that answer. One question from the audience: was the model on a certain area of the cortex or brain, or the entire brain?
So our model geometry is pretty limited. We didn't specify a certain area of the brain. I think it's just pretty generically a cortex. But the neuronal types were borrowed from the original Vertex tool.
I see. Thanks. Hope that answered the question from Catherine. Yeah, we still have maybe time for one or two more discussion points. Maybe, yeah, I see Patrick list a bunch of discussion points he want to discuss. I'm also curious to learn more about that. Yeah, maybe the first question: what are some other ways to analyze the anatomic functional connectivity relationship? Here you show three metrics, I guess, or three ways: the granular causality, the pairwise and stuff, and also the correlation.
Yeah. So I was actually hoping on getting some feedback from the group here. Like right now, we're just doing a very simple correlation between some of these metrics, but I'm interested in like if there are like other ways of doing this, and if there are other like interesting like ways of calculating anatomical connectivity that could be useful for this analysis.
Yeah, okay. That's a bit beyond my knowledge. I'm not sure I can give you some expertise, like feedback on that. But yeah, for people who might know more about this, welcome to talk to Patrick offline to provide him feedback and some further discussion. Yeah, I think maybe one more time, time for one more topic to discuss. Maybe go to yeah, good. Yeah, cool. I'm not sure that there's certain thing you want to mention, but one thing actually confuses me a little bit when I see the analysis is the representation of the functional connectivity. Like I know for anatomic connectivity, you have to weight, which are the parameters or the numbers you can try to analyze with. For the functional thing, is that just LFP? Do you kind of, it's actually not very clear that how you apply this granular causality on LFP signal only. I mean, for anatomic connectivity, you know the structure, you know the weight, you know the edge. Maybe it's more straightforward, but for LFP based functional connectivity, I'm not quite clear how to.
Yeah, so the all the functional connectivity metrics are calculated from the LFP measured in the model. Specifically how they're calculated, full disclaimer, my co-author Julian did the actual calculations, but for like something like granular causality, what it tries to do is it will try to, like at any given point, it looks at how past data from one of the LFP recordings can predict current data for the recording you're interested in. And so using kind of that method, it derives like some connectivity value for a pair of recordings.
I see. Okay, that makes sense actually. Basically try to use the, I see, just you use the LFP from one neuron, try to predict the other thing, the other neuron, try to see whether they have a strong functional correlation from read from LFP or not.
I guess one thing to clarify is like the LFP isn't coming from specific neurons, it's coming from, I guess, like a population of neurons in both in the model and during experimental settings. So
would that be more clear or more specific to individual neuron if you use spike trains instead of LFP?
Yeah, potentially. I think in our study we were kind of interested in like how like metrics from LFP would reflect back to the anatomical connectivity, because LFP seems to be a pretty common way of recording activity in the brain, and so we wanted to see that like the connectivity results coming from LFP actually had some base in anatomical connectivity.
I see. Okay.